visualize it. To do that, the first step, we take our pool of proteins, some of which are actually stuck together in a protein complexes, and denature them by increasing the temperature or adding a chemical denaturant. This breaks up of protein complexes and causes all the individual proteins to unfold. Another function of this denaturation step is to coat each individual protein in a negative charges. So negative charges. In negative charges. Then after doing this, the denaturating step, we load our proteins into a well at the top of an acrylate gel. The gel is actually porous, so it has lots of little holes and spaces inside for proteins to pass through. Then we apply an electric current across the gel, which basically forces the proteins to move towards the bottom, papunta sa bottom. This is why it is really important for the proteins in our sample to all have negative charge, so they are repelled. No? So they will repelled by negative charge at the top of the gel and attracted to the positive charge at the bottom of the gel. Their heights represents the different lengths of sizes of denatured proteins we would use in Western blot. Okay, it is also a loading die that will help us know when to turn off the electric field that makes the protein move. And loading die doesn't stain the proteins or anything. So it just flows the gel ahead of the proteins and indicates that we stop the current. So in Western blot, you want to line up all the proteins according to their sizes or length, because this will make it easier to identify the protein we're looking for. So we know how big are proteins by comparing them to a protein ladder, which we run along the side proteins we got from our cell. A ladder is a solution that contains lots of proteins of known sizes, so at the end of the Western blot, we'll able to compare our proteins of interest to the ladder to determine how big our proteins are. But after running our proteins through the gel, hindi pa rin natin sila makikita. We still can see any of them. So now, we need a way to detect the one we're looking for. So we have the gel, and we have to get the proteins from the gel onto the membrane where we can detect the protein we're looking for. Well, we already made sure that our proteins were coated in negative charges. So we can rely on the same principle of using electric current to move them onto the membrane. So proteins can't just jump through space onto the membrane. So we have to make sure that the membrane and the gel are tightly sandwiched together. We use filter paper and filter pads to keep the gel and the membrane in close contact and coated in a buffer solution. Then, we pack that whole sandwich into a tank filled with a buffer that creates the electric field. So now that we package all our layers that creates the electric field, the proteins are attracted to the positive pole of the tank. So they migrate out of the gel and captured by membrane. So keep in mind that the proteins are still invisible in the eye, hindi pa rin siya nakikita. So we need to visualize the protein we're interested in. So once we have the proteins transferred to the membrane, we will soak the membrane in the primary antibody. So take note for the primary antibody that will only stick to the protein we want to look for. The antibodies are great because they are super specific. So an antibody for protein A, for example, shouldn't bind to protein B. So this gives us the way to tag our protein of interest amongst a large population of proteins on the membrane. So now we've attached the primary antibody to our protein, but didn't make it any more visible than it was before. So that we know that it is actually add another antibody, dito na papasok yung secondary antibody. So in Western blot, there are a few different ways that we can find our proteins or detect on the membrane, and these different methods mainly have to do with some type of secondary antibody that we use, like the fluorophore, no, the fluorophore. We will see if our protein of interest is present and how much protein of interest is present if the thicker band is more if thicker band is more protein, so mas present siya dun sa specific antibody. So that's it po.
But how will you know? There is another machine. What is the purpose of that machine? After you have labeled it with antibodies, how will you know that the protein in it, the protein, how will you know it? That
Once
you have the protein.
Once there will be a band already in the
And how is band formed?
the band form when the second first po is the primary antibody. So if there will be no attachment on it, there will be another called the secondary antibody. Once the antibody is banded in the secondary antibody, so
what is the for how do you know? How will you
the
detect? How will you detect this band?
this band will be detected by the secondary called the fluorophore by the arranged in their height po, their weight.
You cannot detect the band. You have to expose that to where in order that you can detect the band.
Then wait, the question is how to detect the band. So
yes, how will you detect that? Because if you are going to expose that just merely after in the gel, after you run that in the gel, after you have separated it from the gel, you separate it from the gel, and then uh, and then after that,
then the technique
after you have run it to the gel, you run it to the gel.
yes po doc.
Okay, what is the next step after running it to the gel?
So after running to the gel,
Can you see the band right away after running it into
not,
the gel?
not yet po doc.
Oh, how are you going to see that band?
When there will be a attachment po dun sa secondary antibody that is more visible than it was before. So it is doing the add of another antibody na tinatawag pong the secondary antibody.
Is the antibody regulable?
Yes doc.
It is regulable?
Yes
But
doc.
how are you going to expose? How are you going to produce that band? Not anymore from the gel, but rather from the when you transfer it to a cellulose, a paper, you transfer.
Yes po doc.
You show me the first slide. You show me you
This
enlarge this one. No.
the last step po ba doc? The separated
The
protein
last
bond.
step, you have separated the proteins by gel electrophoresis. The question is, when you run it through the electrophoresis, gel electrophoresis, can you see the bond right away?
Sir, after po the secondary band, you can see
After running,
after
that
running
black
in
bands.
the secondary antibody, you can see the bonds after po the attachment of the secondary antibody. And when it is transferred into cellulose to the what do you call this,
Nitrocellulose.
nitrocellulose, doc, you can see it by your naked eye if the band is thicker. If the band is thicker, the more proteins the more proteins is there po.
Ikuan mo nga yung unang slide mo. This is not the one. Yung mayroong ka ding kwan. You have the anong slide yon? Yun. Okay, itong kwan.
Yes, this the last po.
Okay, now.
So the polymer sheet containing the antibody antigen complex is now
Okay,
exposed.
first you have number 2. Is that number 2?
Yes po, the sample of
Okay.
proteins.
Okay, you run your protein in the gel electrophoresis.
Yes po, doc.
Okay? And then, after that, what happened on number 3?
So it separated protein sample is then transferred to a polymer sheet. So the first thing po, before we going to do the gel electrophoresis po, we need to lyse po muna. Then
No. Itong step by step mo, mayroon ka ng step number 3.
Yes,
Then
po,
you
doc.
have step number 4. What happened in step number 4?
After the step number 3 po, the monoclonal antibody, so this is the monoclonal antibody is added onto the polymer sheet containing the separate protein, and it only binds to antigen proteins of interest. So dun lang po muna sa primary antibody po.
So it's not anymore on the gel.
Yes po.
So tinanggal mo ngayon siya, you remove it from the gel now.
Yes
And
po.
then you transfer that to what is this?
After po, you can still see
What is that number 4?
The primary antibody.
What is the material in number 4 where you transfer your protein?
The material that we use to transfer the protein.
Is that the what is that?
The primary antibody po, doc.
Added on the polymer sheet.
Yes po.
Okay. What you did is you have from the gel, you have to transfer that to a polymer sheet.
Yes po.
What is that polymer sheet?
The polymer sheet po is the NSA.
What is that?
The polymer sheet po in Western blood called PV.
The monoclonal antibody is added into the
polymer sheet po.
Polymer sheet. What
Yes
is that?
The
Sheet
PV,
membrane?
the PVD po, the PVDF po, that membranes that the purpose of this PVDF or polynyl difluoride, sorry doc. Polyvinyl
Polyvinyl chloride.
Yes, difluoride po. This
Okay.
This
And then,
Yes po.
na-separate mo the proteins now from the gel was separated.
Yes, Doc. Opo.
And then, it was transferred to the polymer sheet, which is
Yes po.
known as polyvinyl dichloride.
Yes po.
And then after that, what did you do?
So the next step
When
po is
when
yes
it
po.
is already on the poly sheet, what did you do now?
After that po we will going to no after the monoclonal body attachment to the monoclonal body, another antibody is created specific to the
That
first
is the first
monoclonal.
antibody that you
Yes po.
transfer.
Yes po, the
First
primary.
antibody that you transfer, the primary antibody.
Yes
So
po.
the primary antibody binds into the protein.
Yes po.
Then after that, again,
We
another
will do
antibody
another antibody. Yes, yes, Doc. Another antibody, which is what we call the secondary antibody po.
were placed on the sheet.
Yes
And
po.
then what happened to that secondary antibody?
What happened to the secondary antibody is that there will be attachment already of this antibody, and the polymer sheet containing the antibody antigen complex is now exposed to a solution containing the radioactively labeled antibody po.
What happened is that that secondary antibody binds into the primary antibody. That is the definition. It binds into the first thing that happened. The primary antibody binds into the
Secondary.
protein, into the
Protein,
protein, or you call that, you mean the antigen.
Yes po.
It binds into the antigen. An antigen is a protein.
Yes po, Doc. Apo.
This binding. And then after that, the second antibody was again placed or added. And that
Yes.
second antibody binds into the primary antibody.
Yes po.
And that is radiolabeled.
Yes
Okay.
po, Doc.
And then now, what did you do with this shit? In order that you will see the bonds.
The radioactive body will bind to the antibody antigen complexes, and then now it is be visualized on a audiograph. The band will appear on the X-ray film po. So if the protein
But
is
how
present,
will it? How will the band will appear? How will the band will appear? Ay, nandyan sa diagram mo, it is in your diagram. What will you do after that secondary antibody was transferred to the primary antibody? What will you do in order that the band where the antibodies are attached will appear? In the first place, you cannot see all the bands.
yes
Hindi mo
po.
makikita yung bands na yan. Anong gagawin mo ngayon after that?
After the sixth
Nandu.
step po.
So what will you do now after you have put the radio-labeled antibodies, the secondary antibodies? What will you do
Ilalagay po siya dun
in
sa
order that you will see the target protein? Anong gagawin mo? What will you do? Angelo, you are not reading your... Nandyan sa kwan mo, anong ginawa mo dyan sa last step?
the dark band corresponds to protein of interest po. Dun sa X-ray film po do.
Of course, you have to expose that into the X-ray.
Okay.
Expose mo yan sa X-ray ngayon. Upon exposure to the X-ray, that is the time that you will see the band.
Yes.
Tingnan mo yan, isang band na lang yan. how come there's only one band that you can see? And what is that band that you can see? How come there's only one band? Sa lang nakikita mo, there are many bands, ha?
Yes po.
There are many bands, ha? Pinapakita mo, there are 246 bands. 246 bands. But when you expose that to X-ray, there is only one band. Why?
Meaning po, Doc, this is the only protein that is present on the specific study po.
Why it is the only, there are several proteins.
Yes po.
Nakikita mo yung yung sheet mo, nakikita mo, pinapakita mo, ilang bands yan?
If the band is present po, according to the Western blot protocol po, it's only the protein of interest that will appear po at the last stage.
Why? How come?
So because the principle of Western blot is to detect po,
How come it is only one band that appear?
because the Western blot, the principle of the Western blot is to detect a specific antibody po.
That is not the answer, Angelo. Because that band, that single band there, it is where the antibodies, the two antibodies is bound, which is radiolabeled. Yung iba nyo, nakikita mo ba? They are not what?
Radiolabeled po.
There is no antibodies in there.
Yes po.
There are no antibodies. Kaya hindi mo nakita yung 5 bands. You did not see the 5 bands when it was exposed to X-ray. Because for the reason that there is no binding of the radiolabeled antibodies.
Okay.
Kaya sa X-ray, only 1 band was seen, observed, because it is only where your radiolabeled antibodies were bound. Iyan ang nangyari dyan, ang Angel.
Opo.
Huh? You did not read very well the procedure. Kung you didn't even know that in order to expose the band, in order you can observe the band, you have to expose that into the X-ray. Huh?
Na ano lang po, pressure.
Ano ka? Biruin mo ito, 2 weeks na ito ang supposedly ang report mo. Weeks na ito, di mo pa naiintindihan ang gusto. You have not mentioned the chemicals that are used to separate, for the separation of your protein, that will separate your protein, that will make it negatively charged. So that will make your protein negatively charged. What do you mean by SDS, Angel?
Solely
SDS.
SDS po, meaning solely yun.
Di mo alam kung ano?
Page lang po, SDS page. I know po, it separates primary by the mass po. The SDS meaning...
What is SDS page? Anong meaning ng SDS page?
Sodium
That is
yes po.
sodium
Sodium
Dodecyl
dodecyl sulfate polyalkylamide gel po.
polyacrylamide gel
Electrophoresis.
electrophoresis. Ayan ang ibig sabihin. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. That
Yes
is the
po, Doc.
meaning of SDS. No? Masterin mo ito, Angel, you have to understand this very well because when you will be encountering again this Western blot, at least you have an idea how it is being done. Huh?
Yes po, Doc.
Angel?
Yes po, Doc.
Okay. Who is the next reporter?
Doc, si Sir OJ na po.
OG. Paano ka
Hello.
naging OG? Dapat OJJ ka. Oscar? OJJ, hindi OJ. OJG.
For a moment
Okay,
lang
sige,
po.
Oscar. Go ahead, Oscar. What is your report, Oscar?
PCR po, Doc.
Okay. Have you done this?
Yes, Doc.
Okay, Oscar.
Is my slide visible, everyone?
Yes, Sir OJ.
Okay, so, good afternoon to my colleagues and of course, our Professor Dr. Silverio. So, for today, I'll be discussing about polymerase chain reaction. So, particularly, this topic is very familiar to me because I've been working in a molecular laboratory. However, my knowledge is only limited up to the level that I can comprehend. So, at the end of this topic, all comments and suggestions may be um, given at the end.
Anong, Oscar, mga pinagsasabi mo? You have been doing this pagkatapos sasabihin mong limited ang knowledge mo? Paano yan?
I have done the other methods po kasi, Doc. It's only for reverse transcript, RT-PCR po, what we are doing. So,
Yes,
hanggang pa lang
but
po yun.
you have to, after you have prepared everything, you have to use the machine. And the machine will just work.
Okay po, Doc. Noted po. So, for today's discussion, we'll be talking about the inventor of the PCR, which is Carey-Mullis, about also the nucleic acid amplification, and the three steps, major steps in PCR, the pre-PCR, PCR, and the post-PCR. So, to start, so the polymerase chain reaction was invented, or this process was invented by Carey-Mullis, which is a Nobel Prize winner in chemistry at the year 1993. So particularly Dr. Carey-Mullis is a chemist by profession and is also an LSD LSD user. So he is a self-confessed LSD user. However, during this time that he discovered PCR, it was just all of a sudden during a trip going to somewhere. So it just popped out on his memory that he wants to do or he thought about the use of PCR. So
Sir, parang
I just
nag-save, Doc.
Yes, sir. Wala siya.
wala.
Loko. Loko.
Galit na galit.
GG na naman ako. You betrayed me. Sir Alex, oh.
Gusto mo ba i-continue, sir? O wait natin siya? Baka
Hindi,
maulit mo lang kasi. Sir OJ.
Continue ka hanggang sa... Andiyan na po ba? Wala pa.
Wala pa rin, sir eh.
Sir, sa last slide na.
Continue ka sa last slide na.
Try ko
Inalag na.
siya i-message, sir. Baka hindi na na-realize na nawala siya. Wait lang, sir, ha? Baka maulit mo lang kasi kapag nag-continue ka. Okay lang po?
Sige, ma'am. Nag-disclaimer lang ako. Pinagalitan na naman ako.
Ako din. Question kami Sir OJ Sir OJ, paano po yung ganito?
Wakota ka na, Sir Jello. Nahimik na nga lang sa report mo eh.
Ayan na, sir.
Oscar, you continue. You start.
So, again, for the inventor of the polymerase chain reaction, which is a amplification of DNA molecules by applying heat, was discovered by none other than the Nobel Prize winner in chemistry 1993, chemist scientist Dr. Carrie B. Mullis. So, and since then, the use of PCR has contributed so much in medical research as well as in forensic science. So, this particular quote came from Dr. Carrie B. Mullis, in which he said, I think really good science doesn't come from hard work. The striking advances come from people on the fringes of being playful. So it means that mind plays a big role in a discovery of something, especially in science. Okay, so just a brief introduction. So up to date, so during this year, in 2021, it is estimated that the polymerase chain reaction market worth is about $5 billion. So we can see here the trend of the use of PCR. So this is according to the World Population Prospects on the 2019 revision. 1 in every 6 people will be over 65 years of age by 2050. Thus, the rising demand for personalized medicine has thus created tremendous opportunities in field of genetic medicine. So to gain a competitive advantage in emerging field of personalized medicine, biotechnology and pharmaceutical firms are exploring the grounds of genomic medicine and direct-to-consumer genetic testing. So also, PCR is also used to develop these products, and the industry is ramping up the research and development process in the areas of biotechnology where polymerase chain reaction finds its major application. So we can see here the trend of the polymerase chain reaction that in a few years from now, most likely from being a specialized laboratory, I think this could also be included in a routine laboratory. However, this is still a skilled and specialized laboratory. So the nucleic acid amplification, particularly, this process increases the target nucleic acid in a short amount of time. So this is the main features. The main features of nucleic acid amplification is number one, it has high sensitivity and high specificity. This is very important in detection of particularly virus. And then it has also a good turnaround time. It has a short turnaround time, and the detection is only within hours, especially when we are detecting microorganisms. And number three, it is highly versatile. We can use nucleic acid amplification almost in all types of application that we can use. And the nucleic acid amplification can be mainly categorized into two. We have the target amplification and the signal amplification. So in other literatures, there is also called such as probe amplification. However, in many literatures that I have researched, um, the probe amplification is also included as part of the signal amplification. To differentiate the two, target amplification is amplification by enzymatic activity in which it increases the number of target molecules. So in here, in target amplification, the nucleic acid of sequence of interest is the one that is actually replicated. So when it is replicated, it produces millions of copies. While on the other hand, in the signal amplification, what can be easily differentiate this from the target amplification is that in here, signal amplification does not increase the number of copies of the target sequence. However, the large quantities of the signals binds to the target sequence so that the signals become measurable. So again, to differentiate the two, the target amplification um, replicates the target sequence, while signal amplification, it does not replicate the target sequence. However, the signals binds to the target sequence and then become measurable. So these are the examples of target amplification. So under target amplification, we have these following methods. Number one is the polymerase chain reaction, in which I would be talking about this today. Then we also have the ligase chain reaction, the transcription mediated amplification, the strand displacement amplification, and the reverse transcriptase polymerase chain reaction. Well, as for the signal amplification, we have the probe hybridization assay, the branch DNA technologies, and the cleavage-based signals amplification. So again, today we'll be focusing only to the polymerase chain reaction. So in the use of polymerase chain reaction, there is actually a three main process that we do. So in this, we have the pre-PCR, in which this involves the nucleic acid extraction of either DNA or RNA. Whatever the target sequence is, it must first start with a nucleic acid extraction. And then proceeding to the actual analysis or actual amplification, a nucleic acid amplification, which involves three steps, which is the denaturation, annealing, and the elongation process. And after amplification, so we must be able to measure these products by qualitatively measurement or quantitative measurement in the post-PCR process. So let's start in the pre-PCR process. So as I have mentioned earlier, in the pre-PCR process, okay, this involves nucleic acid extraction. So the nucleic acid extraction has three phases. The first phase, or the first step rather, we have the cell lysis. Okay? During the cell lysis, in here, we have here the samples, which is on a test tube. In here, the sample, of course, the cells, the nucleus and the cell are broken upon thus releasing the DNA. So this process involves either mechanical disruption and uses enzymes and detergents like proteinase K to dissolve the cellular proteins and free DNA. So after the disruption of the nucleus, it has now revealed the DNA. So what we do now is the second step. On the second step, in the precipitation, this would now separate the free DNA from cellular debris. So in the precipitation process, it involves use of sodium ions to neutralize any negative charge in the DNA molecules, making them less water soluble and more stable. And then alcohol is then added, which causes the precipitation of DNA from the aqueous solution since it does not dissolve in the alcohol. And then finally, on the last step, since we have precipitated all the cellular debris, or what we call also as um, uh, the substance that we don't want. Okay, my apologies for the loud noise. There's ongoing construction. So upon precipitation of the cellular, rather upon the separation of the free DNA to the cellular debris, we have now, I will now proceed to the purification step. So in here, um, additional removal of the cellular debris or unwanted material will be further be removed. And then, in this step, the DNA is completely purified. So, on this step, it is usually dissolved in water again for convenient storage and handling. So now, on this third step, we already have what we call the eluates. In the eluates, we call this as the actual sample. And it's now ready for PCR. So again, the nucleic acid extraction has three phases. The cell lysis, where the nucleus is disrupted, and then the DNA will come out of the nucleus. And then on the precipitation step, it will separate the DNA from the cellular debris. And then on the purification step, it will remove all the cellular debris and other unwanted materials, and it will purify the DNA. And then we have now a sample, or what we call the purified DNA, or the extract. So in here, as I have mentioned earlier, cell lysis can involve two ways. This can be done in two ways, via chemical and mechanical method. So in chemical method, it can be used by osmotic shock or enzymatic digestion. The enzymatic digestion is the one that we use in the laboratory. In here, we make use of the proteinase K, which which uh, which digest the cell wall. Okay, and then also the use of detergents and alkali treatment can be done in the chemical method. Well, as for the mechanical method, there is a homogenization by the use of blade or pestle. There's also ultrasonication and cavitation, and there's also use of the pressure cell and the ball mill. So whichever of this can be done, okay, but as long as we disrupt the cell wall and release the DNA from the nucleus, and then that's okay. So on the second step, I have already mentioned this earlier. So in the nucleic acid extraction, so um, upon the disruption of the cell wall, so we need to extract the DNA. So in the actual extraction, there are two two ways to do the extraction. So we have the liquid phase and the solid phase extraction. In the liquid phase extraction, we make use of chemicals here to extract the DNA or RNA or the target nucleic acid. So this can be with the use of cesium chloride, gradient centrifugation with lithium bromide. This can also be done with the use of guanidium thiocyanate phenyl chloroform. And this can also be used by Chilex, and also use of alkaline and purification of the poly-A RNA by oligodecellulose chromatography. While as for the solid phase extraction, this can be done with the use of silica matrices or glass particles, diatomaceous earth or diatomites, the use of magnetic beads, base nucleic acid, and then the use of an ion exchange material or the use of cellulose matrix. So if you're gonna ask me which of this is the one that we use, in our laboratory we make use of the magnetic beads um, as the main method for extraction of the nucleic acid. So aside from that, there are also devices that are used in the extraction. So what are these? So we have the spin columns, the magnetic beads that I have mentioned earlier. These are usual. The spin columns and the magnetic beads can be done manually. So there's manual extraction. However, since we are already moving forward and being modern, there is already this is much preferred because it's quicker and it's less to errors. And also the use of microfluidics and lab-on-chip cartridges. Okay. So this is what happens during extraction. So here we have our sample. Okay. So we have our sample which will be lyse. Okay. Then it will disrupt the cell wall. And then during the binding and washing phase, so this is the one that we call the precipitation phase. So in the binding phase, okay, the the extraction um, the extraction medium, okay, will bind to the target DNA. So then in the washing phase, okay, the cellular debris would be washed out. So in here, this is the use of the spin column. So we can see here that what remains on the top is um the actual the extracted nucleic acid. While when it is washed, so the cellular debris will come out. Okay, so it's like a filter. So it will be filtered so that on the top part of this spin column, what will be remain here is the extracted nucleic acids. And then as was mentioned earlier, we need to further purify the um, the the extract. So there's another washing that will happen. So in here, another washing will happen to further purify the sample. And then after another washing, so we can now have the purified nucleic acid. So we have here the eluates or the elute that we call, and this will be now used as the sample. Okay. So this one is the magnetic in the use of magnetic beads. So we can see here that it has beads here. So the beads will actually bind to the nucleic acid, thus leaving the cellular debris floating, which then after washing will be discarded. And then what remains here are the magnetic beads with the nucleic acids. And then after washing again, so what will remain now is the purified nucleic acids. So I have here a sample video on how we will isolate RNA from a tissue or a cell. This one is the manual extraction. So this one is with the use of trizol agent. It's chemical method, the liquid phase rather. Like, okay.
Oscar, there is no sound.
Wala po talaga siyang sound, Doc. So on that particular method, they make use of the chemical or the liquid phase extraction. On this next video, I'll be showing how to do it on the solid phase with the use of the magnetic beads. So again, this one is the one that we use in the laboratory. So again, once you have extracted the nucleic acids, this is now the one that we will be using for or will be running in the PCR machine or in the thermocycler that we call. Okay? So on the next step, so in the actual polymerase chain reaction, so there is now an amplification of the DNA. So not only DNA can be done or can be amplified in the polymerase chain reaction, but RNA can also be amplified in polymerase chain reaction. However, there's a special step in the amplification of RNA. So it will be first transcribed into DNA so that um, it can be replicated via the it can be replicated in the PCR machine. So the applications, polymerase chain reaction is used to obtain DNA, of course, that can be used for cloning genes or in phylogenetic studies. It also can be used to identify microorganisms. This can also be done for DNA fingerprinting, detection of antibiotic resistance, and detection of virulence genes, and etc. This can also use for the detection of biomarkers and single nucleotide polymorphisms, or what we call SNPs. As I have mentioned earlier, in the polymerase chain reaction, there is another three-step process. In this three-step process, there is a varying temperature. So, this three-step process, the denaturation, the annealing, and the elongation involves different temperature. So, on the denaturation process, this is set to a high temperature between 94 degrees Celsius to 96 degrees Celsius. While in the annealing, the temperature is set between 40 to 60 degrees Celsius, while the elongation process, it sets at 72 degrees Celsius. So, why is there a high temperature in the denaturation step? So, for the DNA to here to unwind, so the denaturation step first is the DNA is needed to unwind. So, we all know that we needed rather high temperature denatures proteins. So on that same principle, this is applied to the DNA. So for it to be unwinded, it needs high temperature. And then on the second step, which is on the annealing, so after the unwinding of the DNA, primers will then bind to the target sequence. Okay? Primers will bind to the target sequence. The same with the DNA polymerase. So after annealing, once the primers and DNA polymerase binds to the target or the starting point of the replication, on the step 3, which is elongation, the DNA polymerase now will extend the gene, making an exact copy of the target sequence. So in here we can see this, in this video we can see the process. For the first part, it was the denaturation process. On this step, it is the annealing part where the primers and the RNA polymerase attach, and then what is going on now is the elongation process where the nucleotides now will attach to its base pair here, thus forming an exact copy of the other one. Thus the replication happens. So in summary, we can see here that in the polymerase chain reaction, in the denaturation process, again, it will unwind the double helical structure of the DNA, thus exposing a site for the primers and DNA polymerase to bind. So once the DNA primers and DNA polymerase bind to the starting point, it will now start to synthesize the lagging strand. So that after which, on the elongation part, it will produce a new copies, and then these copies now would undergo the same 3 steps again. That will produce 4 copies, and then these 4 copies will produce 8 copies, and then these 8 copies will undergo another cycle, continuing 20 to 30 cycles, and some can reach up to 45 cycles depending on your protocol, and we will now be able to produce millions and millions of copies of the DNA. Thus, we call these products as amplicons. Okay, again, we call the products of the polymerase chain reaction as amplicons. So going back, the products of the nucleic acid extractions, we call them as LU8s. Okay? So, when we talked about the polymerase chain reaction, so we have talked about the temperatures. Now we talk about what is the reagent that is being used in the polymerase reaction. So in this, there is what we call the Pcr Master mix, or the Pcr mix that we call. This mix is composed of different components. The first one is the DNA template. the DNA template, this is the one that we have extracted. So this is our LUA. Okay? Next one is our DNA primer. This one, it has primer 1 and primer 2, in which of course, the primer 1 is designed as the starting point of the leading strand, and the primer 2 is designed as the starting point of the lagging strand for it to be simultaneously replicated or synthesized. And then we have the DNA polymerase and the DNTPs, the magnesium, which is the one that um, prevents other interferencing substances from disrupting our process. And then we have the PCR buffer, which is the one that maintains the pH of the solution. So the DNA polymerase, so this is the enzyme that attaches the DNTPs to the primer. So a special requirement for DNA polymerase that it must be a heat stable DNA, because as was mentioned during the denaturation step, a high temperature is involved. So a good DNA polymerase is needed. So what is commonly used is the TAC polymerase, which came from Thermus aquaticus, this one. But other alternatives can also be used, such as the VEM and the PIFU. The VEM came from the Thermococcus littoralis, and PIFU came from the Pyrococcus furiosus. So these are all heat stable DNA polymerases. So, as I have mentioned earlier, magnesium is needed, particularly the magnesium chloride. So, what does it do? So, for it to be oriented, okay, properly oriented during extension, magnesium chloride is the one that helps it. So, it forms soluble complexes with the DNTPs as the building blocks. So, this one influenced primer annealing, strand dissociation, formation of primer-dimer artifacts, and enzyme activity and fidelity. So, as I have mentioned, magnesium can also prevent other interferencing substances from influencing the PCR reaction, because the presence of hemoglobin, ADTA, heparin, and other chelators, okay, will affect the PCR reaction. Okay. And then we have the buffers in the DNTP. So the buffers, this provides the suitable environment for the polymerase chain reaction to take place. And the DNTPs, these are your dinucleotides. So you have here the AGTC, which binds to this target sequence in the elongation process. So the most common used buffer is the tris chloride. Okay. So upon amplification, okay, we need to measure the product. So we have the amplicons. So however, there are two types of measurement of the product. So in here, we have the endpoint and the real time. On the endpoint, it was called endpoint because you need to finish the whole polymerase chain reaction before we can actually measure the number of amplicons. Okay? However, still, on the endpoint detection, it will not give you an exact number of the amplicons or amplified nucleic acids, but it will just give you an estimate of the amplicons. So on the endpoint, this uses the agarose gel electrophoresis, that is also called as the closed method, open method rather, because it happens in a gel electrophoresis, and it uses toxic and carcinogenic dyes such as titanium bromide. That is why endpoint PCR is not commonly used nowadays. However, still, this can also be used, but not most clinicians prefer it because, again, of toxicity, and carcinogenicity. So, on the other hand, the real-time PCR gives a real-time detection. So what happens here? While the PCR is running, or while the polymerase chain reaction is happening, we can monitor the process. If there is an actual amplification and there is no amplification. So what is advantage of real-time over endpoint is you do not need to finish the whole PCR reaction or the actual process before you can know that there is a amplification. Okay? Because there is also no need for gel visualization, and it uses safer fluorescent dyes such as the Cyber Green, the FAM, ROS, Texas Red, and Big dyes. Okay? While the measurement of real time give us a relative and absolute count. So how do we know this one? So it gives us a value we call the cycle threshold value, or the CT value, which gives us a relative and absolute value of the nucleic acid that was amplified. So start with the endpoint. So in the endpoint, as I have mentioned, it involves the agar gel electrophoresis. So in the agarose gel electrophoresis, you need the following components. Of course, number one is you need the agarose powder. So the agarose powder is needed to make the agarose gel, which would be the of um, main medium of this method. And then the buffer where the agarose gel will be suspended. And of course, the electrophoresis setup in which we need the electricity. So in here, the agarose powder. So there is a specific concentration of agarose depending on the type of DNA molecules that we want to measure. So the DNA molecules have following weight, the molecular size rather. So if we wanted to measure DNA molecules with the size of 5,000 to 60,000, then this concentration is recommended. So the lower the concentration, the larger the DNA molecules it is able to capture. And the higher the concentration of the agarose gel, the lower or the smaller the DNA molecule it can measure. So buffers, as I have mentioned earlier, this is the one that we use either for suspension or for tracing the migration. So we have the loading buffer and the electrophoresis buffer. The loading buffer tracks the migration, while the electrophoresis buffer is the ionic salts that is the one that we use as a suspension medium for the agarose gel. While the ethidium bromide is an intercalating agent and associates with the base pairs of the DNA strand. So I have mentioned earlier, this one is not, the endpoint is not recommended for most because again, it is highly toxic and carcinogenic because it's also known, it is a known mutagen to humans. So also with we need to make use of the ultraviolet transilluminator so we can see the actual bands that are present in the sample. So how do we read the bands? Okay. So how do the the gel electrophoresis happen? So we see here that this one is the whole setup of the gel electrophoresis setup. So we can see here that in here we have the agarose gel, and then we have here buffer float. Okay, and then we have the power supply, which gives up the current. So on my left side, we have the cathode, and on my right side, we have the anode. Okay? So we can see here that The sample will be then loaded into the wells, okay, on the wells, and it is said that DNA have a negative charge. So when the power supply gives off an electricity, the negatively charged DNA will migrate, of course, towards the opposite direction. So from negative, it will migrate towards the positive. So we can see here that there is already a migration of the DNA. So the the nearer to the cathode or to the well, the higher the molecular weight of the analyte it is. And the farther to the well, the lower the molecular weight of the analyte. So this means it is smaller in size, okay, when it reaches the farthest from the well. So so this is how we read it. So we have here in an electrophoretic or an agarose gel, once viewed in a UV trans illuminator, we have here a marker. So what is the purpose of this marker? This will serve as our reference from the number of base pairs that we have. So we know that genes have a specific number of base pairs. So depending on the type of gene that we want to detect or the target gene that we want to detect. Like for example, I wanted to detect a gene that has 800 base pairs. So the samples will be loaded into the well, and then electric currents will be induced, and then the genes will be then, the gene fragments will then be, the nucleic acid fragment will then migrate. So on sample A, we can see here that there is no DNA fragment that was detected on the 800 base pair. Thus, we can know here that the target sequence that we are looking for on the sample A, it's not present. However, in the sample B, there is a band, there is a visible band shown in the 800 base pair or the 800 BP, thus saying that our sample B has the presence of the target gene that we wanted to detect. While as for the sample C, we can also see here that there is an absence of the band on the 800 BP, thus saying that sample C does not have the target gene that we are looking for. So among these three samples, only sample B has the target sequence. So that is when we know that there is an positive result for sample B. Like for example, only an example, like the gene that is supposed to code for spike protein is under 800 BP. So we can see here that sample A is negative for COVID. Sample B is positive for COVID since it has band present in the 800 BP, while sample C has negative result for COVID because it does not have a band formation in the 800 BP. So that is how we interpret the band formation or in the gel electrophoresis, the presence in absence of band. Okay.
Juan, Oscar,
Yes, Doc.
but if you're talking of marker, these are the these are the target DNA.
Yes, this is the Doc, the reference
The,
po.
the reference DNA. And this indicates, like for example, if we want to look at the presence or if a person is contaminated with COVID-19, that means that one of these markers refer to that to the DNA or to the RNA of COVID-19.
Yes, Doc. So for example, Doc, as I have mentioned earlier, this one is just an example, Doc. So
Yeah.
for example, the gene, the spike protein, has 800 base pairs. So all the samples that will have a band formation on the 800 BP will show a positive result for COVID po.
But you are talking here of DNA.
Yes, Doc. The gene, rather, the DNA that we...
The
that is
DNA, you are not
supposed to-
talking here of spike protein.
No,
You are-
the DNA, Doc. The DNA that codes for the gene that codes for the spike protein.
Okay. Okay, that's the DNA.
Yes po. Should I proceed po?
But, okay, that is only a fragment of the
Yes.
entire DNA of the virus or the microorganism.
Yes, Doc.
That is only
A
one
fragment
part.
of the gene that codes for the...
Yeah, that is only a certain fragment, because if it's coding for a spike, that is only one part of the virus, the spike.
Yes, there
But
are
there
more...
are other components or parts of the virus
Yes.
that is being coded.
Yes, Doc.
So you have to understand, class, that it is the DNA that is being measured in here. And then previous to that, it was detected that this 800 base pair is the DNA that is coding for the spike. Or this is the another name for spike, attachment, which is responsible for the attachment to the target cell. Okay. So that is every protein that is being coded as different as different size, and it has different size, molecular size.
Yes, Doc.
Okay, you have to be very clear on that, okay? Or this is the ladder DNA. We call that as the ladder DNA, DNA ladder. That is the DNA ladder. Okay? So, okay, continue, Oscar.
So moving on, so that is how we measure the on the endpoint method. Well, when we go to the real-time PCR, so in here, this is also known as the kinetic PCR or the homogeneous PCR, whereas amplicons are assayed as they accumulate after each cycle. So we can see here that we can measure the number of amplicons after every cycle because it will reflect on the display monitor that can be seen here on the number of amplicons that will be measured. And it does not use agarose gel, and it's a closed tube system, thus preventing contamination from external substances. So this also shows quantitative measurements, such as this can be good in the quantitative measurements, particularly in the viral load of the Hepa B virus, HIV virus, Hepa C virus, cytomegalovirus, and BCR AVL. Okay, so how do we measure now, or how do we detect the products or amplicons in the real-time PCR? So there are different detection platforms that can be applied to real-time PCR. We have the 5' nuclease assay. We have the double probe FRET. FRET stands for fluorescence, resonance, energy transfer. Okay, and can also be via molecular beacons. Can also be used by a scorpion and cider green. So let's enumerate. Let's understand what happens on these different platforms. So on the first platform, the 5 nuclease assay. So we can see here the Tachman probe. So we make use of a probe here. So on this probe, it has a reporter dye and a quencher dye rather. On this part, once the probe binds to the target DNA, okay, it will bind to the target DNA, and when the primer and DNA polymerase extends, it will now cleave off the, apologies for a moment. Okay, going back again, we have here in the 5 nuclease assay, we have here a probe which has a reporter dye and a quencher dye. When this reporter dye and quencher dye is still together, it will not emit a fluorescence. Okay? But during the elongation process or the extension process, when the primers and DNA polymerase elongates, along the way, it will, of course, meet the reporter dye. And then the reporter dye will be cleaved off, of course, along with the other nucleotides, leaving the the quenching dye on the end. And when the reporter dye cleaved off in the probe, it will actually give off a fluorescence. Thus, this one is the one that is being measured in the 5 nuclease assay or in the PCR detection system. Okay. On the other assay, or in the other method, whereas dual probe FRET or the fluorescence resonance energy transfer, we have here a donor probe and an acceptor probe. If these two are together, it will not emit any fluorescence. However, once the annealing stage or the extension process begins again, okay, as the as the primer and DNA polymerase extends throughout the template, so it will cleave off the donor probe. Okay, thus this donor probe have already donated the energy to the acceptor probe, and this acceptor probe now will fluoresce, thus showing an activity, and this fluorescence activity is the one that will be measured now. Okay. On the other method, in the molecular beacon method, so in here we have a molecular beacon probe. So the molecular beacon probe looks like a stem, okay, whereas here the fluorophore and the quencher are still together. Once this probe binds to the target hybrid, it will, from a stem-like projection, it will flatten and be laid off to the target DNA. And then the fluorophore now will be unattached to the quencher. And this fluorophore without quencher will now show fluorescence. And this fluorescence now will be the one again to be measured in the detection system. Okay? On this method, the scorpion timer, we have here a scorpion probe. Okay? So in here we can see here that there's also a fluorophore and a quencher. So once the probe binds to the um, target DNA, okay, it will start to elongate. Okay. So what happens here in the elongation process? We know that there is a heat that is involved there. Okay. So with the heat, this projection will start to change, going from this like lollipop shape projection going to this scorpion-like projection that it is called as a scorpion primer. Okay, so now once this quencher is far away from the fluorophore will emit an exciting light, thus producing fluorescence again, and this fluorescence will now be then measured in the detection system of the PCR. Okay. Lastly, we have the cyber green. This one is the easiest to understand. Why? Because cybergreen will only bind to a double-stranded DNA. So if it's denaturated, so there is no binding of cybergreen that we can see here because there is no double strand here. Okay? Only single strand. While along the extension stage, so we can see here, or the primer annealing, the primer is attached to the starting point, thus we can already see a we can also already see the cybergreen attaching to the double strand here on this part. And upon the completion of the extension phase, we can see here that the cybergreen have attached to all of the double strand, to the whole double strand, thus emitting fluorescence. Okay? Only when cybergreen attaches to the double-stranded, only then it will emit fluorescence. So if it is unbound, so it will not emit any fluorescence. But if it is bound to a double-stranded DNA, it will now emit fluorescence. Thus, that fluorescence will now be then measured in the detection system of the PCR.
Oscar, in that
Yes,
case, there are several cyber green in the mixture.
yes, Doc.
It's not only two, it's not only one, but there are several in the mixture.
Yes, Doc.
Okay. So in that case, so in other words, there will be the synthesis or the elongation will occur only if there is cybergreen attached.
No, Doc.
Attached.
After the elongation process, the cybergreen will attach to the double-stranded because the cybergreen only bounds to
Okay,
double-strand.
bounce into the double strand. Okay. So so now
If
then
there is,
what will happen to the cybergreen?
it will emit fluorescence when it is bounded
There will be a fluorescence. Okay, there will be illumination. And so this illumination will be picked up by what?
by the detection system of the PCR, Doc.
Detection system with the PCR. Okay. Okay.
Okay. So, how do we measure these fluorescence? So, we need to quantify them. So, in the quantification, this is expressed as an absolute value, either copies per ml or IU per ml. So, in the quantification, we make use of this standard curve, okay, to measure or to quantify the amount of amplicons or amplification that happens. So how do we quantify it? So in here, we have an amplification plot that we can see here. So on this part, we have the number of cycles. And then on this part, we have the threshold line. So for it to become so we can say that it is amplified, it needs to pass through the threshold line. So we can see here that there are four amplifications that happen on this particular cycle. Okay? So in here, to measure the amplification or to have the actual value, we will determine the CT value on the point where it started to amplify above the threshold line. Okay? So on this part, like for example, just an example again. So the red one is the FAM. Okay, it's the FAM dye. The yellow one is the Vic dye. Okay, the green one is the Sci-5 dye, and the blue one will be the Rox dye. This is just an example again. Okay, FAM, Vic, Sci-5, and the Rox. So each of this dye, okay, represents a DNA fragment. Okay, represents a DNA fragment that was amplified. So for example, the DNA fragment that the FAM, rather the DNA fragment. Okay, so we have, for example, we have 4 target DNA fragments. So once these are all present, so that means that there is a amplification. But if there is no curve above the threshold line, that means that there is no amplification that happened. So, for example, there are only 3 or 4 rather, there are only 3 um, amplification on this amplification curve. So that means 1 is absent. So for example, here, please bear with me. We have FAM, we have the ROX and the Sci5 and the VIC. So this, for example, the FAM, ROX, and VIC are the dyes that binds to the DNA fragment that codes for the gene for COVID. Again, I'm using the example for COVID because this is the one that we do. So, for example, again, we have FAM, ROX, and the VIC gene. So the FAM, ROX, and VIC gene, okay, will be binds to the DNA fragment that codes for COVID. Okay, while the Sci5 rather the Sci5 dye codes for the internal control. So to simply put, if there is only the presence of Sci5 here, there is no FAM, ROX, and VIC, then that means it is negative for COVID. While if there is an amplification like this with complete presence of the dyes, then that means it is positive for COVID. So something like that. So why do we need to have an internal control? Of course, for me to be able to establish a criteria if the result is accepted or rejected. So, for example, again, in here, we have the FAM, VIC, ROX, and the Sci5. The FAM, ROX, and VIC stands for are attaches to the DNA fragments that codes for the spike protein, okay, of COVID-19. While the Sci5, this one is the internal control that we use to decide whether the test run is accepted or rejected. So again, if there is only presence of the internal control, then that means it is negative for COVID. But if there is presence of all dyes in the amplification curve, then that means it is positive for
Oscar, the internal control, is it the marker?
no, Doc. It depends on your sample that is being used. Like for example, in
Is
our
that the sample? Is that the sample? The internal control, is it a sample?
the marker of the sample, Doc. Like for example, in our institution, the internal control or the internal control is the N gene, Doc, that codes for the human. It codes for a certain human gene to specify whether it's an oropharyngeal or nasopharyngeal sample, Doc.
But it is still, but it is not the DNA or
It's
a
still a
COVID
DNA
DNA,
fragment
but
of
it
that
is not a
human
COVID
gene.
DNA.
It's not a COVID DNA, Doc.
It is not.
It is not po.
Okay. Of course, since it is not a COVID DNA, so it's negative.
It's negative, Doc. So, but
Yeah.
still, we
It's
need to
negative.
have those internal control.
If without the red and the green dye, without the red and the green dye, of course it is negative
Yes, Doc.
because the indicator of a COVID presence of COVID nucleic acid is the red and the green dye.
Yes,
Is
Doc.
it?
Yes, Doc.
Okay.
But
Okay.
if there is absence of all dye here,
Yeah.
so we can know that the test run is actually invalid. So we need
The test is
the test run is invalid, Doc, because
invalid. Okay.
amplification
Because
of the internal
yes.
control.
Yes, because of the internal control, which is supposed to be amplified.
Yes, Doc.
Okay, it is natural that if the red dye and the green dye is absent, that means there is no nucleic acid of the COVID.
Yes, Doc.
Yeah. But since it is the marker, it has to be present in there
Yes, Doc.
in your diagram during the in your amplification diagram. Okay. Continue, Oscar.
So that is how we interpret the amplification curve. Again, if the internal control is only present, then that means, like for example in COVID, it is negative for COVID. But if the other reporter dies, rather the other dies are present aside from the internal control die, then that means it is positive for COVID. While if all samples are, if all dials are not present on the amplification curve, then we can say that the test run is actually invalid. But what if we have the presence of the red and green, the FAM and ROX, but we don't have the presence of IC? So what would be the deciding factor here? So this is still to be considered as a invalid run. So there are few factors that contributes to the invalid to the validity of the run. Maybe because the master mic was not prepared properly, or there are other or the temperature profile was not suitable for that certain sample. So still there are still many things to consider on that part. So but then again, we use the amplification curve again to measure the amount of DNA fragment that is present on the sample. So the amplification curve that I have mentioned, since in the real-time PCR, this can be viewed while the PCR is running. Thus, it is called as the real-time PCRs because you can always view this while PCR is running. Unlike in the endpoint, you can only view the results once the PCR has been done, and you'll only measure the products. Okay? So, aside from the amplification curve, if we use other probes, okay, such as the cyber green, okay, or FRET probe, then we can also make use of the melting curve analysis. So we can see here that this is not the cycle, okay, but rather the temperature. As I have mentioned earlier, okay, if we want to measure the amount of signals that is present, then we make use of this. So in here, the melting temperature is is reflected at which 50% of DNA is double-stranded and 50% is melted or single-stranded. So as we go back, cyber green only attaches to double-stranded DNA. So that means there is a complete amplification during that part.
Oscar,
Yes.
just clarification that this is why we are green. This is fragment of nucleic acid. This
No,
is a nucleic acid.
this cyber green po is a dye din po.
It is a dye.
Yes, Doc. A specific oligonucleotide probe rather. It's a probe.
Or sequence. It is a dye or
With,
sequence. It is a sequence. Huh?
yes,
Uses
Doc.
either SBR green or
A sequence
sequence.
specific, Doc.
Is
Either,
that a DNA sequence or a
either
nucleic acid sequence?
if
Huh?
you use cyber green, which is a dye, or an sequence specific oligonucleotide probe. So you can make use of
Ah,
either of it, Doc.
so it is on. It is just a dye. It
Yes,
is
Doc.
not a nucleic acid.
Yes, Doc.
It is not an oligonucleotide probe.
Yes, Doc.
Okay. Okay. So the main purpose of that is just for illumination.
Yes, Doc, because we can see the reflection of the melting temperature shows us where at which 50% of the DNA has completed elongation, thus forming a double-stranded structure po, which is the one that is needed for cybergreen to bind po.
Okay. Okay. So it is just dye. All the green, the red, all those are dyes.
Yes, Doc.
Okay. But that dye, is it radioactive?
Fluorescence, Doc.
It just fluoresces, but it
Yes,
is not radioactive.
yes, Doc.
Okay. Unlike if it is a sequence, it could be, if it is an oligonucleotide probe, it has to be
Yes,
radiolabeled.
yes, Doc.
Ah,
Yes po.
okay.
So,
Continue, Oscar.
okay. So we can see here on this particular temperature or melting curve that on this part, on the 80 degrees, okay, there is already a formation of, or the 50% of the DNA that was formed have already became double-stranded. Thus, we can see here that there is an actual fluorescence on that temperature. Okay. So, Um, so we have understood the process of the polymerase chain reaction from nucleic acid extraction to actual nucleic acid amplification and the post-PCR analysis or the analysis or the measurement of the amplified products. So, well, since we know all of this, so the application of PCR can be applied in a um, uh, wide variety of applications, particularly in personalized medicine. Personalized medicine, this refers to pharmacogenomics and pharmacogenetics, while for personalized nutrition, it involves the nutrigenomics and nutrigenetics. And this can also be applied in genetics or epigenetics. We can sequence, we can um, perform, um, yes, we do sequencing of the genes, and then in the global transcriptomics is we compare the presence of, or rather we develop here a whole transcriptome of the genes that can be present um, in our surroundings. Other, it can either be a human gene, it can either be a plant gene, animal gene, or other types of genes. So these are the reference that I have used for this particular topic. And
Okay.
if you have questions or comments, that you
How
are,
about the RT?
I'm
Because
sorry, Doc.
if we are talking, Oscar, of the nucleic acid of a virus, it is RNA.
Yes, Doc. Um,
RNA.
on this part,
So
here, Doc. So, on that special, ano po, like for example, for COVID, since it's an RNA virus, so there is no DNA that is present on the viral particle. So, what we need is we need first to, we need to add an additional step in this process. We need to first transcribe the DNA, and the RNA into DNA.
but it is included in the mixture
Yes, Doc.
or in the cocktail. That
The
what is that
Pcr Master.
enzyme again? Involved in the conversion of DNA, of RNA to DNA. What is that enzyme again?
Um, for a moment, Doc.
Anybody from the class? Raquel.
Reverse transcriptase enzyme group.
Of course, you should know that, Oscar. That is the reverse transcriptase. It is reverse transcriptase because if you are talking of HIV virus, the HIV virus itself contains reverse transcriptase in the genome. It is reverse transcriptase. Okay, Oscar,
Yes, Doc.
that is a good presentation of PCR.
Thank you, Doc.
Okay, saan ang laboratory mo again, Oscar? Where is your laboratory, molecular lab?
It's located here in Mandaluyong po. The Pcr Molecular Diagnostics po.
Is that a private
Yes,
lab?
Doc.
And how much are you charging for every PCR?
It depends, Doc. But for routine PCR, for SARS-CoV, we charge for 2,500 something, Doc.
2,500. And the results will be taken, the
24
results
hours, within
24 hours. 24 hours. Okay, class, you have to understand this very well about how to operate or how to perform PCR. Like for example, Oscar, this is an added value for him to have knowledge of this PCR. How much is your salary, Oscar? Nakakakuan ba yan? Can it support 3 families?
One family only, though.
One family. How much are you receiving now?
Around, ano po, Doc, 40,000 or something.
40,000. It's okay.
Yes.
Because if it's not, but the problem is that your job in there is not stable, unlike if you are in the government.
Yes, Doc. That's the only problem.
You apply now. The military now is looking for medical technologies that have knowledge in the operation of molecular laboratory, meaning RT-PCR. You go to that laboratory, you apply to that, and there is a lot of privileges. Oscar?
Yes, Doc.
They are in need. The military now, PC, Armed Forces of the Philippines, they are even the Coast Guard. I have students, graduate students, who are doing their masteral, working in the Coast Guard, working in the PNP, Philippine National Police Laboratory.
Yes,
Their
po, Doc.
molecular laboratory. You have to apply on that, Oscar, with your knowledge, with your knowledge of PCR. Sa PNP, send your application. You will have a stable job. Okay? Who is the next reporter?
Doc, si Miss Zora na po.
Sora. Vincent, are you ready, Vincent?
Yes, here.
Okay, that's good, Sora. Nice to hear your voice.
Putangin na. Lagi siya walang masyadong tanong sa akin.
What is the title of your
Never.
presentation, Sora?
Akala ko
It's about HIV.
Okay. Vincent, are you ready?
Just let me try to share my screen.
Okay.
Can you see it?
Yes, ma'am.
Wait. All right. So, Our topic is about the role of current drugs used against HIV. For the overview is 1. Immunodeficiency due to HIV infection. 2. The main preventive measure is to adopt safe sex. 3. Advanced stage of the disease can be complicated by severe infection and malignancy. 4. There is no cure and the progression can be controlled with medication. As we can see,
Sora, can you make it in slide form? Your presentation that is not slide form. Can you make it in slide form?
wait. 喂 Oh, wait a moment.
他说什么？
Vincent, make the presentation in slide form.
"没有你的那个 PPT， 等一下。
没有我的 PPT 啊。
不是你的 PPT。我是全屏放了呀，但是
不是，就是你让它弄成那种播放模式就可以。
我是按照播放模式的呀，我在共享屏幕。
要把麦关了。
Oh, all right.
Oh, no. Sora, can you not make it? Make it in slide form.
不是，你用鼠标右击。
Sir, is okay? Sir.
Yes, ma'am, that's okay.
Oh, okay, thank you. So the first is about the AIDS overview. Part 1 is immunodeficiency due to HIV infection. 2 is the main preventive measure is to adopt safe sex. 3 is advanced stages of the disease can be... What?
Just continue, ma'am.
Okay. Thanks. 3 is about advanced stages of the disease can be complicated by severe infection and malignancy. 4 is there is no cure about progression can be controlled with medication. As we can see the picture below, the left part is about the symptom of the AIDS, and the right part is what the AIDS virus look like and the way it express and transform. So first we should know what the AIDS is. AIDS, also known as the acquired immune deficiency syndrome, is a systemic disease caused by human immunodeficiency virus, HIV, also known as the AIDS virus. HIV infection can lead to different degrees of immune deficiency in the human body. Untreated, the infected people in the late stage of the disease is prone to complicated with a variety of serious infections and malignant tumors, and eventually lead to death. This disease is infectious. So the root about the HIV's transmission is HIV mainly exists in the blood, semen, vaginal secretions, cerebrospinal fluids, pleural and abdominal fluids, etc. And milk and other body fluids of the source of infection, and its infection and transmission canals is include like sexual contact and blood products and transmission by mother to child, just as this picture had showed us. So, statistics released by the Chinese Centers for Disease Control and Prevention show that by the end of 2020, a total of 1,053,000 people in China had been infected with HIV, and 351,000 deaths had been reported. The proportion of this transmission grew to 74% and 23.3% in 2020, respectively. Men who have sex with men are the group at highest risk of contracting HIV. Older men in China are becoming increasingly infected with HIV, with a proportion of new HIV positive cases among men aged 60 and above, rising from 7.41% in 2010 to 18.21% in 2020, mostly through sexual contact, according to the Chinese Center for AIDS and STD Prevention and Control. So, drug therapy like DAA. At present, there are a total of 6 categories of more than 30 drugs. Just include like compound preparations can treat AIDS, representatively for nucleoside reverse transcriptase inhibitors and like non-nucleoside reverse transcriptase inhibitors, and also like PIS, like INSTIS, also like FIS and CCR5 inhibitors. So in present, there are a total of 6 categories of more than 30 drugs can treat AIDS. Representatively for NRTIS and NRTIS and PIS like INSTIs and FIs and CCR5 inhibitor. So there are these kind of drugs in China, and there is mostly just like HAART, a combination of antiviral drugs and viral therapy for adults and adolescents is shown in the table below. This is a table about some data we can see here. It's about the recommended program. And each English for abbreviations in Chinese like this. Then is the principles of prophylaxis after HIV exposure treatment regimens. 这是个啥呀？ Like TTF or FTC plus RAL or DTG are recommended, and depending on the local resources, PI such as LPVI and DRVI can be used if INSTIS is not available, and also like start time and duration of treatment. Medication should be administered as soon as possible after exposure to HIV, preferably not often, not more than 1 day, but even after 1 day is recommended. The course of medication was continuous administration for 28 days. Next is about the anti-HIV drugs and their treatment options. It's because of the spread of HIV can cause a disease, HIV or AIDS. Potentials are often very negative. Because AIDS is incurable, discouraged. AIDS has now been developed anti-AIDS drugs. By taking anti-AIDS drugs, can let the AIDS patients live like normal people. Anti-HIV drugs and the drugs used in their treatment regimens act on the reverse transcript of the HIV virus, known as that like antiretroviral drugs or reverse transcriptase inhibitors. People who know about AIDS know that the AIDS virus is the antivirus genes of the retrovirus family, and its spread needs to be catalyzed by the reverse transcriptase, which exists in the center of the virus. Another class of the drug acts on viral protease. Proteases is also an indispensable catalyst in the HIV replication process, and its role is mainly to catalyze the maturation of the new replication of the virus. These drugs inhibit the activity of proteases and are called anti-protease drugs or protease inhibitors. Principles of anti-AIDS drugs. The principle of the anti-AIDS drugs is to interfere with the virus steps of HIV replication in the human body, so that the HIV into the body cannot multiply normally. In other words, the drugs do not actually kill the virus itself, but only protect the body from further infections. HIV is a retrovirus, and its replication needs to go through the following steps. First, bind to the receptor on the surface of CD4 plus T cells and NK cells. Second, under the action of reverse transcriptase, reverse transcription begins, synthesizing DNA from RNA. Third, the synthetic DNA is integrated into the host cell genome. Fourth, under the action of RNA, polymerase transcription subsists of new mRNA. Fifth, translate mRNA to synthesize new viral proteins. Sixth, new viral protein and new RNA assembly to form new virus particles. 7. Release new viral particles to infect more CD4 plus T cells. And then stopping just one of these 7 steps can have an anti-HIV effect. Anti-HIV drugs are different from broad-spectrum antivirals, which are drugs that specifically target HIV. In the case of membrane fusion inhibitors, the lipid vesicle members of the vascular virus is basically the same structure as the host cell member. So, membrane fusion inhibitors do not work on the lipid, but inhibit on the surface of the viral vesicle membranes or the separator on the surface of the host cell membranes, thus preventing the glycophoration and the receptor from binding to initiate the membrane fusion process and making it possible for the virus to enter the cell. Therefore, membrane fusion inhibitors can only work on specific receptors, thus inhibiting the corresponding viruses from entire the target cells, which is very specific and usually can only be applied to one or a few viral infections and cannot be a broad-spectrum antiviral. The target cell of HIV are CD4 plus T cells. The receptors is CD4 molecule, and the co-receptor is CCR5 or CXCR4, which means that when HIV uses its GP120 protein as a key to unlock the T cells, it needs to bind both the receptor and the co-receptor to initiate the process of membrane fusion into the cell. Either CD4 and CCR5 or CD4 and CXCR4 anti-HIV membrane fusion inhibitors that enter either work against the HIV glycosylation GP120 or GP41, or against the receptor ligands CCR5 or CXCR4 of CD4 plus T cells. Then it's about the prognosis. HIV, also called AIDS, cannot be cured at present, and active and effective treatment can help HIV patients delay the progression of the disease. However, if patients cannot tolerate drugs or fail to take drugs on time and consistently, the immune deficiency of the infected patients will be further aggravated, and the progression of to AIDS will be accelerated, leading to serious complications and they believe that AIDS is incurable. However, if it is found early and standardized treatment is carried out in time, the life expectancy of AIDS patients can be close to the normal population. Then it's about the harmful of the AIDS. AIDS can cause severe immune deficiency in the human body, which is prone to bacteria, virus, fungal, parasitic, and other infections. Due to without immune resistance, the infection will be harder to cure and can lead to death. Similarly, due to immune deficiency, AIDS can make people more susceptible to malignancy, which can lead to death. Because AIDS can be transmitted through sex and blood, coupled with social fear of AIDS, will make the life of AIDS patients, marriage, work, social, and many other aspects are subject to great restrictions. Then, complication. Opportunistic infection disease like bacterial, viral, protozoal infection, and other opportunistic infections, and eventually the patient may die due to system failure. All kinds of opportunistic tumors is an important cause of eventual death in AIDS patients. So, what should we do? How can we prevent it? But there is a current no effective HIV-based vacuum that can prevent HIV infection or slow the progression of AIDS. Then maybe just cutting off the transmission of AIDS is an important way. Yeah, that is an important way to prevent and control AIDS. Like the specific ways to cut off transmission. Here are some examples, like cut off the sexual transmission of HIV, use condoms currently, and protect safe sex, like no drug, no sharing of noodles, avoid extrinsic transmission of AIDS, avoid unnecessary injections and blood transfusions during medical procedures, prevent mother-to-child transmission of HIV in maternal pregnancy examination. HIV infection, regardless of the number of CD4 plus T lymphocytes, should be given appropriate antiviral drugs before delivery treatment and fetal preventive treatment to play a certain role in blocking the transmission of HIV. The risk of HIV infection in high-risk individuals can be reduced by taking antiviral drugs in combination with safe sexual practices known as pre-exposure prophylaxis, PrEP. Thank you. That's all my presentation.
Thank you, Sora, for the report. Sora, is Vincent around? Sora.
The real Vincent is absent, but the fake one is here.
Somebody is requesting, Sora, somebody is requesting to see your face. Can you present your face? Somebody, Alexander, wants to see your face in the... Can you show your face, Sora?
Why?
Yeah, because somebody is requesting from your classmates.
So just let me ask Zora if she wants to share her face.
Are you another student? You are not Sora.
Yeah.
Oh, you are Sora.
I'm the fake Vincent.
Okay. Oh, okay. Can you show us your face? Because, you know, is it possible?
You want to see who, Sora or this fake Vincent?
No, your face, Sora. You show us your face in the slide or in the laptop.
他让你好像 show your face， 看你的脸，真的假的？你自己回答一下吧。你回答一下 how 或者说 why， 你就那个说一句嘛，就让大家看一下你的。可以。可以。你就说 you're here， 就是 I'm here 嘛，然后你就那个。他好像想看你的脸，我也不知道。Zora 叫你呢，没事，你就随便说一句话吧 ，Zora， 或者你开一下你的视频 ，Zora。Yes
Okay, can we see now your face, Sora?
你就开个视频让大家看一眼你的脸。
现在吗？
对对对，你现在开一下吧，就看一眼，打个招呼，然后没事没事没事。
我打开摄像头了，好像它不露脸。
我关掉，等一下，我停止共享试试，可以吗？
我再试试。不可以，为什么都不露脸？
She don't know how to show her face.
Hi, Satoman. Okay.
我已经，你告诉他。
Can you enlarge it,
哦
the whole face, the entire face, Sora? Oscar is very happy to see your face, and Alexander.
什么？你给我翻译，我这边。我操！你告诉我，我这句话怎么说？就是 person 的是教授是吗？ Hello, person.
Your entire face, can you move your laptop so that we can see your entire face, Sora?
全脸，哎呦！
Okay, we can see it now. Oh, Oscar, what can you say about Sora? She is still very young. How old are you, Sora? How old are you?
Oh, 23.
23. Okay,
Yeah.
Alexander, where are you, Alexander? Okay, can you see now the entire face of Sora? Okay.
Yes, po, Doc.
How about you, Peter? Okay, can you see now the entire face of Sora? Sora is still young. Okay. By the way, Peter is asking if you have a boyfriend, Sora.
Yes.
Do you have a
Yes.
boyfriend, Sora? Huh? Sorry, Oscar.
Yes.
Sorry, Peter. Sora is already taken.
什么？你继续跟我说。
Okay. Now, about your report, Sora, it has something to do with HIV. Okay, you have started on how the HIV infect a person, infect a cell.
我能关上了吗？
The HIV virus really needs a receptor in order that it could infect the cell. You must understand, class, that the HIV virus contain an enzyme. It has a built-in enzyme known as reverse transcriptase. And this enzyme is responsible for converting RNA to DNA. And remember also, class, that the virus, it is a retrovirus, meaning that it contains, its nucleic acid is RNA. And so, what will happen when the virus infect the cell? It has to attach, it has to bind to a receptor of the target cell or the host cell. And then, by endocytosis, the virus enter the cell. And then, what will happen is its covering, okay, is removed. Is removed, and then it releases the RNA in the cytoplasm. It releases the RNA into cytoplasm. But the problem is that the RNA could not be inserted into the chromosomal DNA of the host cell. And so the RNA has to be converted to DNA. So the RNA now will enter the nucleus, it will enter the nucleus, then it will be converted to DNA. And then that is the time that the DNA, viral DNA, will be integrated into the chromosomal DNA of the host cell. And the enzyme responsible for this integration is known as the enzyme integrase. It is known as the enzyme integrase. And once it is integrated in there, the DNA will start to be transcribed into messenger RNA, and then the messenger RNA will be translated into protein. And the translation process, or it could be translated, or it could be synthesized into protein because of the presence of an enzyme known as protease. The enzyme known as protease. So, in that case, when the RNA is translated into protein, it develops now the functional structure of the virus. Like, for example, what are these functional structures of the virus? Like, for example, the capsid. Like for instance, the covering. The covering. And these are all made up of protein. And then, what will happen now? The RNA, again, it will be packaged by this functional protein to make another virus, to develop another virus. And this virus, they are known as virions. And these virions will be responsible for the lysis of the cell, and then it will release the new virions, and then once it is outside the cell, it will infect other healthy cells. And that is how infection occurs. But now what Sura is discussing also is about the drugs of choice for the treatment of AIDS. And most of them, they are known as inhibitory drugs, inhibitors. These drugs inhibit the enzyme. It inhibits the enzyme, these drugs. What do I mean by it inhibit the enzymes? It inhibits the three enzymes responsible for the growth and development of the virus. First, the integrase. The inhibitor, this drug inhibitor, will bind into the enzyme integrase. We know the principle of the principle of inhibition. Okay? We know the principle of inhibition. The inhibitors bind into the binding site of the enzyme. And so what will happen if the inhibitor will bind into the enzyme integrase? If that is the case, the DNA of the virus could not be integrated into the chromosomal DNA of the host cell. That is one of the action of the drug inhibitor of the enzyme integrase. Another inhibitor is responsible for the inhibition of the enzyme reverse transcriptase. The inhibitory drug binds into the reverse transcriptase, and so the RNA could not be converted into a DNA when the inhibitor is bound into the enzyme reverse transcriptase. And so, with that, again, the DNA, there will be no transcription of the DNA to messenger RNA. And so there will be no translation into protein. Another inhibitor is involved in the inhibition of protease, enzyme protease. When the enzyme protease is inhibited, the structural components of the virus could not be synthesized, like the capsid, the spike, the covering, the viral envelope could not be synthesized because the enzyme protease is inhibited. So these are the three main actions of these drugs, HIV drugs. They are in the form of inhibitor. And because of this, the virus could not multiply. The virus could not divide. The virus could not use the operation. The operation cannot control the operation of the nucleus. Okay, so this is what Sora was reporting. And of course, the mode of infection, how this virus is infect human, it is by body fluids and other forms of human action, by drugs, injection, blood transfusion. This is how the virus is transmitted. Okay? Is there any reporter?
Sir, we have 2 reporters left.
We have still 2 reporters left.
Yes,
I think
sir.
they are going to report next meeting.
Depende
next
po sa inyo,
Saturday.
Doc.
Who are the last reporters?
Sir Peter and Ma'am Sol.
So what is your report, Peter?
Doc, I'll be reporting on conjugation, transduction, and transformation.
Okay, the 3. How about you? Who is the other one?
Ma'am Sol, sir.
Sol, what is your report?
Doc, I will be reporting about DNA profiling.
DNA
fingerprinting, Doc.
DNA fingerprinting. Okay. So are you okay? Our last meeting will be next Saturday.
Okay po.
So you are going to report. You will be reporting next meeting. And then during our last meeting, we will also discuss how the final exam will be given to you. And again, all the reports must be in. You have to upload that on the Google. And also, I also posted an assignment in the classroom, Google Classroom. And some of you have already answered the assignment, and I am expecting that. Okay, do you have any question, class? Do you have any questions? So, I want to see you next meeting before the Christmas break. Huwag muna kayong maging excited. Do not be excited. Sora.
Yes, yes.
How is Christmas now in China? Is it white Christmas?
你告诉我怎么回答。
Is it now snowing?
你直接说，你直接说。
Is it very cold?
No, actually, there is no snowing, and the weather is still hot.
The weather is still hot. Okay. Okay, so it's still
In the southern part is still sunny day and the temperature is high, but in the west,
still high.
Yeah.
Western part it's already cold.
Yeah.
Okay. Sora. Okay, see you next meeting. Unless you have any question, we're going to terminate our class today. Okay?